So research question, right? I would say this project in some ways is kind of questionless.
When I was just starting my lab, I had a very strong interest in this genomics approach called TNSeq.
It's a sort of tool building thing that was going to let us answer other questions down the line.
And the idea that I could do an experiment that would allow me to identify all of the genes that were contributing to any particular trait is almost too good to be true.
But at its most basic, what it let us do is understand what the repertoire of essential genes was in the genome.
And I met Blake, and he felt similarly.
I had started my career and my PhD working on a pathogen called Cryptococcus neoformans, a fungal pathogen that infects people typically that are immunocompromised. And despite the fact that many people have not heard of Cryptococcus, it's the AIDS-defining illness for the majority of AIDS patients. It's also uniformly fatal if left untreated. One of the things that's really fascinating about it is that they're incredibly similar to humans. Very closely related evolutionarily. One of the things that had sort of frustrated me as a PhD student was that we didn't have a lot of tools to work on Cryptococcus. Of the 7,000 or so genes in the Cryptococcus genome, some of those are things that are absolutely required for that pathogen to grow, and some of those things are relatively dispensable. But if you develop a new drug that inhibits one of these things that's absolutely required for growth, it can kill the pathogen.
The novelty of Blake's work was not in the TNSeq approach itself. Blake's big accomplishment was adapting that approach in this very important fungal pathogen. It gives us another organism in which we know all of the genes that are essential for life and establish the protocol for building a big mutant library. So this is a great service to the Cryptococcus community, and he's identified genes that affect Cryptococcus's susceptibility to an important antifungal drug, fluconazole.
The traditional method is to basically delete one gene at a time.
Thousands of times over and over again, and each mutant is represented in that collection, frozen in a tube like this.
And in contrast, TNSeq lets us make deletions for the entire genome.
Rather than being stored in thousands of little tubes like this, they can then be stored in one little tube.
One of the things that's kind of unusual about fungal pathogens is that most fungi don't grow at human body temperatures. This actually happened in Cryptococcus at some point about 100 million years ago. So how do you become a pathogen? How do you evolve a pathogen? Cryptococcus is fairly ubiquitous in the environment, so you likely inhale some number of yeast cells of Cryptococcus in any given period of time. If you're healthy, you clear that very quickly. If you're not, it can set up residence in your lungs and cause pneumonia. It will then move from the lungs into the brain. That's the predominant cause of fungal meningitis. That's absolutely fatal if left untreated. It has a predilection for growing off of some of the sugars that are in the brain, and so it's essentially eating your brain.
Many of the genes that are essential in Cryptococcus, most of them, are also conserved in humans and very likely to be essential for life in humans as well.
It's one of the things that makes them so hard to treat. This is a step forward for us towards better tools to help us develop new antifungals and new treatments for very, very dangerous diseases.